In a groundbreaking development, scientists have harnessed the power of generative artificial intelligence to create novel antibiotics, starting from the tiniest of building blocks, a single atom. This revolutionary approach could potentially arm humanity with new weapons in the ongoing battle against antibiotic-resistant bacteria, one of the most formidable adversaries we face today. Imagine a world where we can outsmart superbugs that have evolved to resist our current arsenal of antibiotics. That's exactly what a team of researchers, led by Dr. James Collins, the synthetic biologist at the Broad Institute of MIT and Harvard, has achieved. Their work, published in the prestigious journal Cell, details the creation of two new antibiotics, NG1 and DN1, which have shown remarkable effectiveness against multidrug-resistant bacteria, including the notorious gonorrhea and MRSA, methicillin-resistant Staphylococcus aureus. But here's where it gets controversial. The traditional methods of antibiotic discovery have been painstaking and often fruitless. The new generative AI models, however, have not only discovered but also designed these antibiotics from scratch. This leap in technology could redefine how we approach antibiotic development, potentially saving countless lives. Collins, who co-founded FairBio, a nonprofit dedicated to antibiotic development, is now working to bring these promising candidates to clinical trials. And this is the part most people miss. The process of designing these antibiotics involved training AI models on vast chemical libraries. The first model, used to create NG1, was trained on compounds known to be effective against gonorrhea. It analyzed chemical fragments and predicted their antibacterial activity, identifying a potent structure called F1 as a starting point. The model then iteratively added elements, assessing the resulting compound's potential to combat bacteria at each step. The second model, which produced DN1, took a different approach, building new compounds from a single atom, essentially decorating it with additional elements and bonds while predicting the compound's antibiotic potential and toxicity. The real challenge, however, lies in synthesizing these AI-designed compounds. While the models can predict the likelihood of synthesis, the actual process is the main hurdle. Collins' team collaborated with Ukrainian manufacturing partner Enamine to synthesize 24 of the identified candidates, 7 of which showed antibiotic effects, including NG1 and DN1. The team's success in this area could pave the way for more groups, including pharmaceutical companies and governments, to adopt this technique and develop much-needed new antibiotics. The implications of this research are profound. Collins envisions a future where the development of 10 to 15 new antibiotics could significantly improve our ability to combat antibiotic-resistant bacteria. This is not an insurmountable goal, especially considering the resources available to nation-states and wealthy philanthropists. The public availability of the code for these new models invites collaboration and innovation, potentially accelerating the discovery of new antibiotics and saving lives around the globe.